from us. It's gonna be like one-to-one, very private counseling sessions. So in case you need any extra help apart from your studies, you can also discuss that with our team. It's gonna be more like a friendly buddy session. So you can unload your burden with us. Now we have three different subscription plans: basic, standard, and premium, starting from 15K and going up to 50K. And we have different facilities available. In the premium, you'll have live doubt clarification as well, one-to-one personalized guidance for you also, along with the notes. By using my coupon as displayed on the screen, you can get extra 20% off on the available subscription plans. Okay. So let's begin for today's topic, that is GAGs, glycosaminoglycans. As usual, let's take a clinical case scenario so that it builds up interest for you. A 3-year-old male presents with facial coarsening, joint stiffness, and hepatosplenomegaly. Parents report frequent upper respiratory infections. Physical examination reveals corneal clouding and a short stature. Radiographic evaluation shows dystostosis multiplex. Based on these findings, what is the most likely diagnosis? Okay. Once we're done with the GAGs and mucopolysaccharidosis, you'll be able to answer this. Moving ahead, let's begin with the introduction. So glycosaminoglycans are negatively charged heteropolysaccharide chains. Okay, we've already discussed what is heteropolysaccharide. That is different sugar units when joined together to form a polymer. It is known as heteropolysaccharide. And when the sugar units are same, it is known as homopolysaccharide. So your GAGs are negatively charged. Now, glycosaminoglycans. So it has glycos in its name. So glyco, you must be having an idea that it's something related to carbohydrate. Amino is something related to amino sugar, and glycan is a synonym for polysaccharide. You study chemistry of carbohydrates. So, and also that your GAGs are heteropolysaccharides. So your GAGs are found abundantly in mucin. So they have another name known as mucopolysaccharide, and it was first seen in mucin. So also it contributes to this name, mucopolysaccharide. So GAGs are also known as mucopolysaccharide. They are found abundantly in mucin, and it was first elucidated in mucin. Now moving on to the structure of GAGs. GAGs are long, unbranched heteropolysaccharide with repeating disaccharide unit. Now, this must be reminding you of something we've already studied, glycogen. So as you move forward, please don't forget what you've already studied. In contrast to glycogen, we had discussed that glycogen was a branched homopolysaccharide made up of alpha D glucose. Now, GAGs are unbranched heteropolysaccharide. So here the repeating unit is a disaccharide unit because it is a heteropolysaccharide. So that is two different sugars are repeating itself. Now, that repeating disaccharide unit, it consists of amino sugar and an acidic sugar. Now that amino sugar can either be glucosamine or it can be galactosamine. Now what is the difference between glucosamine and galactosamine? Both glucosamine and galactosamine are 6-carbon aldose sugar. That is, they are both hexoses, okay? Now, what is the difference? That is, they are epimers at carbon 4. That is, at carbon 4, the orientation of hydroxyl and hydrogen is different. So, that is the only difference between your glucosamine and galactosamine. That is actually the difference between glucose and galactose. Now, how glucose and galactose are changed into glucosamine and galactosamine? That is when the hydroxyl at the second carbon is replaced with an amine group here. You can see that is how a glucosamine or a galactosamine is formed. So in GAGs, the disaccharide unit has an amino sugar. So how is that amino sugar formed? By replacing the hydroxyl at carbon 2 with an amino group. And they are of two types, glucosamine and galactosamine. Now acidic sugar. Now acidic sugars which can be present is glucuronic acid and iduronic acid. Firstly, let's talk about that what are acidic sugars, okay? So acidic sugars, they are formed when the farthest hydroxyl, that is at carbon 6, is oxidized to COH, a carboxylic acid. So this is your acidic sugar. Now glucuronic acid, okay, I'm continuing then. So the other part of the disaccharide unit is acidic sugars. Now your acidic sugar, it is glucuronic acid or iduronic acid. How are your acidic sugars formed? They are formed when the hydroxyl at the farthest from the carbonyl group, that is hydroxyl at the carbon 6 is oxidized to carboxylic acid. So that is your acidic sugar. Now glucuronic acid, this is glucuronic acid, this is iduronic acid. They are epimers at carbon 5. This is your carbon 5 here. Carbon 5. You can see that when the carboxylic acid group is above the plane, it is your glucuronic acid, and when it is below the plane, it is iduronic acid. So they are epimers at carbon 5. Now your GAGs are composed of amino sugar and acidic sugar. Now amino sugar can be glucosamine or galactosamine, or the acidic sugar can be glucuronic acid or iduronic acid. So it will form a disaccharide unit which looks something like this. This is the disaccharide unit. You can see here, this is the acidic sugar. And which one is this? Glucuronic acid. And here you can see amino sugar. The hydroxyl has been replaced by an amino group. So this is the amino sugar. And here you can see the NH2, one hydrogen has been replaced by an acetyl group. So this is acetylated, okay? Your amino sugar has been acetylated. Now this disaccharide unit that is formed, it is repeated. It can be repeated n number of times to form what is known as GAG. So this disaccharide unit is repeated multiple number of times to form GAGs. So I hope the structure of GAG is clear. This is it. Okay. Now properties of GAGs. Your GAGs are negatively charged because it has acidic sugar. So your acidic sugar has this. So this gives it the negative charge which contains the carboxyl group. Okay. That are negatively charged at physiological pH. And it has amino sugar which is acetylated or sulfated. Okay, so in some cases it is acetylated and sometimes it is sulfated. So both of this gives it a negative charge. So negative charge is accumulating in the gap. Now, gags repel each other because of the accumulating negative charges. Obviously, the negative charges, they repel each other just like the like poles of a magnet which repel each other. The gags are also having, all of them are having negative charges which repel each other. So when brought together, this slip past each other. This produces the slippery consistency of mucus secretions and synovial fluid which is rich in gas. So your mucus, you know that mucus is very slimy and slippery. It is because it is rich in gags. And why do gags slip past each other? Because they have negative charges which repel each other. So gags are negatively charged, so they attract water. Thus, gags are very good at absorbing water. So the gags, they absorb water and swell up. When the water is squeezed out, they occupy a smaller volume. And when compression is released, they go back to the original hydrated volume because of the repulsion of negative charges. So this property is known as compressibility or resilient. And gags are resilient, therefore seen in synovial fluid and vitreous humor. This can also be explained with the help of this diagram. See, now your gags, they have absorbed water and they have swollen up, okay? Now on compression, this water is removed. This water goes away. But when you again hydrate it, it absorbs water on removal of the compression and it goes back to its original form. So you see, when there is compression, the water is removed. On removal of that compression, it again absorbs water and goes back to its original hydrated form. So gags have this property of absorbing water. Okay. Now classification of gags. According to repeated disaccharide unit, type of glycosidic linkages and degree and location of sulfide groups. So according to the disaccharide unit, that which amino sugar is present, which acidic sugar is present, or on the type of glycosidic linkages and the degree and location of sulfate groups, that is how many and where the sulfate units are present. They are divided into various types, and the six major types, we're going to talk about them one by one. So your first one being chondroitin sulfates. 4 and 6, it only shows where the sulfate groups are present, the position. Okay. Now the disaccharide unit N-acetyl galactosamine with sulfate on either carbon 4 or 6 and glucuronic acid. Okay, so let's see the structure. The disaccharide unit in your chondroitin sulfate, it is composed of N-acetyl galactosamine. So this is your galactosamine and you can see that it is acetylated. This is your acetyl group. Okay. And here you have glucuronic acid as the acidic sugar. And where you can have sulfates at 1, 2, 3, 4. So it says either at carbon 4 or at carbon 6. This is your carbon 6. So this is what forms your chondroitin sulfate. It can have either this structure or this structure. Either your sulfate can be at carbon 4 or it can be at carbon 6, and it is acetylated. Okay, now this is the most abundant gag found in cartilages, tendons, bones, and aorta. Now, moving on to the next major gag, that is your keratin sulfate. You get asked about these gags in or heteropolysaccharides in short notes, okay? So you must know these. Keratin sulfate, disaccharide unit here is acetyl in acetyl glucosamine. You can see the acetyl group here. And you can see this is a glucosamine and galactose. Now, this is the keratin sulfate is the only gag which does not have uronic acid. It has a galactose, okay? So this is important. This is the only gag which does not have a uronic acid. Sulfate content is variable and may be present on carbon 6 of either sugar. So sulfate can either be present on galactose or it can either be present on N-acetyl glucosamine. Now there are two types of keratin sulfate, 1 and 2. The 1 is found in cornea and the type 2 is found in loose connective tissues. Now comes your dermatan sulfate. This is type 3. Disaccharide unit here is N-acetyl galactosamine. Okay, this is again acetylated N-acetyl galactosamine, and here you have hydrochloric acid. And the sulfur is found on carbon 4. It is found in skin, blood vessels, and heart valves. Heparin. Disaccharide unit in heparin is glucosamine here, and glucuronic or hydruronic acid. So it can be either of the two acids can be present. And you can see here that this glucosamine, it is not acetylated. Most of the glucosamine residues are sulfated, and sulfate is also found on carbon 3 and or carbon 6 of glucosamine and carbon 2 of uronic acid. Okay, so most of till now we have studied that all your glucosamines were or galactosamines were they were acetylated, but this one most of the heparins glucosamine residues they are sulfated. Okay, and sulfate is also found in carbon 3 and carbon 6 or on carbon 2 of uronic acid. Now here uh, both the acids can also be present, either glucuronic or hydruronic acid. This is the only intracellular gas. Okay, other gags are all extracellular, but this is the only intracellular gag. It is present inside mast cells and it is an anticoagulant. So that was about heparin. Now your heparin sulfate, it has the same disaccharide unit as your heparin, except that your glucosamines are acetylated and there are fewer sulfate groups. This is an extracellular gag found in basement membrane. Moving on to hyaluronic acid. Disaccharide unit here is N-acetyl glucosamine. You can see this. This is acetylated. This is your acetyl group. So N-acetyl glucosamine and glucuronic acid. All gags except for hyaluronic acid are sulfated. So this is the only unsulfated gag. As you can see, there are no sulfate groups here. All the other gags, they were sulfated. But this hyaluronic acid is the only gag which is not sulfated. And also, it is not covalently attached to protein. Now, we'll just talk about this, what it means that it is not attached to protein. And only gap not limited to animal tissues, that is, it is also found in bacteria. So there are a lot of exceptions in hyaluronic acid. First, that it is the only unsulfated gas. Secondly, it is not attached to any protein. And then it is also found in bacteria. It serves as lubricant and shock absorber, found in synovial fluids of joints, vitreous humor of the eye, the umbilical cord, and loose connective tissue and cartilage. Now, let's talk about proteoglycans. What are proteoglycans? They are found in the ECM, extracellular matrix, and outer surface of the cell. There is a central core protein to which approximately 100 linear chain of GAGs are covalently attached. These chains may be composed of approximately 200 disaccharide units. So what is it trying to say? That there is a core protein. This is, let's suppose that this is your central core protein, and to this core protein, okay, I'll draw it here. There's a core protein, and to this central core protein, approximately how many? 100 GAGs. 100 GAGs can be connected, and each of these GAGs that are covalently attached, they can have approximately 200 disaccharide residues or units. Okay, the linkage between the protein and the carbohydrate chain is through a trihexoside. Now here, this linkage, it's talking about this linkage. This was your protein, central protein. This is your GAG. So GAGs are covalently attached to your central protein. So this attachment here, this linkage, it's talking about this covalent linkage. This is through a 3-hexose unit, trihexoside unit, which is composed of galactose, and xylulose, and a serine residue respectively. An O-glycosidic bond is formed between xylulose and hydroxyl of serine. So you know that your serine, it has a citron which has hydroxyl. So the hydroxyl of that serine, it forms a O-glycosidic bond with the xylulose, and that is how your GAG is attached to the protein. So these proteoglycans, they have a bottle brush shape. GAGs are erect, extending out from the core of the protein as they repel each other due to the negative charges. So they are not clumped together because they have negative charges, so they repel each other. So they are extending out from the core protein and they are erect, thus giving it a bottle brush shape. Now your proteoglycans, the protein content is very low. It is only 5%, and your GAG content is 95%. Whereas there's another thing called glycoproteins, in which the protein content is very high, that is 95%, and the carbohydrate content is very low, that is only 5%. An example of your glycoprotein can be antibodies. Okay. So let's see the structure of proteoglycans with the help of a diagram. This here. This here is the central core protein. To this central core protein, what is attached? GAGs are attached. Okay, these are your GAGs that are attached. How many GAGs can be attached? Approximately up to 100. And what is the length of the GAGs? The length of the GAGs is approximately 200 disaccharide units. Okay, this is the top view of the GAG. Now let's see the linkage. This was your core protein here. Now this core protein, how is it attached? The serine. The serine side chain, which has a hydroxyl group, it forms an O-glycosidic bond with xylulose here. And xylulose, galactose, and galactose. This is the linkage region here. Now, to this galactose, your repeating disaccharide units of GAGs will be attached. So this is the core protein. This is the O-glycosidic linkage, and then you have the linkage region, and to this linkage region, your gag is attached. So that was about the structure of proteoglycans. Now, synthesis of gags. The synthesis of gags occur in endoplasmic reticulum and Golgi apparatus, and degradation, it occurs in lysosomes. Defect in degradation of GAG constitute a particular disorder known as mucopolysaccharidosis. So your GAGs, they are degraded inside the lysosome by the lysosomal hydrolases, and if there is any defect in the degradation of these GAGs, it gives rise to a group of disorders which is known as mucopolysaccharidosis. Now, moving on to mucopolysaccharidosis, they are a hereditary disease caused by deficiency of any one of the lysosomal hydrolases involved in the degradation of GAGs. This leads to intralysosomal accumulation of GAGs. What is intralysosomal? That is inside lysosome. So your GAGs are accumulated inside lysosomes as they are not being degraded. This leads to intralysosomal accumulation of GAGs in various tissues, causing a range of symptoms. So all your mucopolysaccharidosis, they are autosomal recessive disorders except Hunter syndrome, which is X-linked. Okay. So there are various types of mucopolysaccharidosis, but the most studied and the most important is Hurlers and Hunters, and that is what we are going to talk about here today. So in Hurler syndrome, your enzyme deficiency is alpha L iduronidase deficiency. So there is accumulation of heparin sulfate and dermatan sulfate. Now, what are the features? There is corneal clouding. Now try to remember that clinical case scenario, okay? Developmental disability, there's dwarfing, there's coarse facial features, upper airway obstruction, hearing loss, deposition in coronary artery leads to ischemia and early death. And in treatment, you have enzyme replacement therapy available. So that was your Hurler syndrome. Moving on to Hunter syndrome, the enzyme deficiency here is iduronate sulfatase deficiency. The degradation of dermatan sulfate and heparin sulfate is affected. So there is intralysosomal accumulation of dermatan sulfate and heparin sulfate. It is X-linked disorder, therefore it is only affecting the males, and all others were autosomal recessive disorders. So you see, only your Hunters is X-linked and it affects only males, therefore. There's clear vision and no corneal clouding. This is in contrast to Hurlers where there was corneal clouding. Okay. And in treatment, you have enzyme replacement therapy available. So that was for today's topic. It was a short topic, but you very frequently get asked about writing a short note on GAGs. So it's important from that point of view. If you have any queries, you can ask. So we'll be taking up revision after this class. So also on the revision class, if you have any doubts, you can come up with your doubts and have a clarifying session. And there will also be revision and recapitulation of what we've studied till now in carbohydrate metabolism before we move on to our next series on lipid metabolism. So thank you for watching.